We're here in SOHO in Houston, Texas, and my talk is going to focus on the role of immunotherapy in acute myelogenous leukemia, specifically the role of BiTEs, DARTs, and ADCs, which are antibody drug conjugates. And my interest in this has been, over the past decade, we've been trying to develop targeting approaches for AML. And this is a much more difficult project than targeting ALL. In ALL, there's several good targets, and one can see that not only are they good targets highly expressed on malignant cells, but they're also expressed on those cells only and on B cells. And so when you target those malignant cells, you're able to kill the malignant cells, but you also kill the B cells, but people can live without B cells. So in general, it's well tolerated, highly effective. Now, when you move to myeloid malignancies, it's a completely different issue. The main issue is that the targets are not ideal. So there are targets that are overexpressed in myeloid malignancies, but they're unfortunately overexpressed in normal hematopoietic stem cells. So anything you would do to target AML will also, in theory, affect the viability and function of normal stem cells, and you need your normal stem cells to produce blood cells. And the other more important issue is that those same targets are also expressed on maturing myeloid cells, such as monocytes and neutrophils, and therefore you have this enormous sink of target existing in the non-malignant mature myeloid population, which you have to get past to get to the malignant AML cells. And these myeloid cells also have suppressive properties, and so they suppress any immune response. So you have a huge sink, and you have suppressive biology, and the targets are also expressed in hematopoietic stem cells, making immunotherapy for AML incredibly complicated. So what are the approaches? The approaches are first to develop proof of principle preclinical models. People have done this, targeting known antigens, shared antigens such as CD33, CD123, CLL1, for instance, and to show that they work in a preclinical mouse model, which people have done using CARs or BiTEs or DARTs and ADCs, and then to take that forward to the first clinical trials, which is where we are right now. So where we are in the development of immunotherapy and targeting therapies for AML is we've identified targets. They're not ideal, unfortunately, and they're not specific to AML, which makes this even more challenging. Number two is that we haven't figured out how to overcome the suppressive environment of AML cells and the monocytes that surround them. And number three, this big sink of target in the non-malignant population is difficult to get past. And the fourth challenge is that the expression of targets on monocytes and granulocytes is really problematic for immunotherapy, because those are the cells that we know now are very much involved in cytokine release syndrome. And so we have seen exactly what you would expect from those challenges. Number one is that all immunotherapies have had modest benefit. There is proof of principle that they work in a human setting, but the response rates are not as great as one would like. That's a problem. Number two is that, and this is a consistent problem no matter what kind of immunotherapy you're talking about, and that is that there is a large percentage of patients that develop cytokine release syndrome, which limits the use of these agents. And so the future is going to be threefold. Number one, to define new targets, and that may be difficult. In other words, there may not be any really specific targets to an AML cell that are not expressed on a monocyte or a normal hematopoietic stem cell. That's a real possibility. And so we're going to be left with trying to target the antigens that are currently available in such a way as to make them more specific. So I think the first approach to find new targets is going to be very challenging. It's possible, and it might require a very exhaustive proteomic approach, genomic approach. The genomic approaches so far have largely failed, but obviously there may be proteins and things that are expressed on the surface that might be expressed at very low levels at the transcriptional level, but are novel and on the surface of AML cells that define them in some way. I'm not optimistic, however. And then the other is to enhance the activity of those immunotherapies to make them work better, since the response rates are low. So to make them work better, people are trying all sorts of stuff. And the problem with making them work better is that there might be the other dreaded complication of worsening cytokine release syndrome, which would limit their activity. So people are using checkpoint inhibitors to make them work better. And then other groups, like our group, are trying to develop mitigation strategies for CRS that would block CRS to allow the escalation of the dose of these drugs so that we can get by that sink and that barrier of target on non-malignant cells to get to the malignant cells. And the third, I think, the third challenge, and this will be, we have to start thinking outside the box for AML. Maybe immunotherapies that are really inferior and don't work very well can be enhanced in some specific way by targeting the sensitivity of a leukemic cell to immunotherapy. In other words, you could give a low dose of an immunotherapy that you think will not work very well, but then enhance the sensitivity of the AML cells selectively to those immunotherapies. So that's one approach. It would be like using things like BCL2 inhibitors and MCL1 inhibitors to allow prevent those cells from fending off the cytotoxic effects of immunotherapy. And the other approach is to figure out a way to actually target two antigens at the same time. So when you target two antigens, suddenly you can then start to look at really selective targeting of AML. So there are several antigens that would specifically identify either a leukemic cell or a stem cell in a leukemic cell, and it wouldn't include monocytes or granulocytes or maturing myeloid cells. So figuring out how to actually target two targets at the same time so that the only time you activate the immune system is when both targets are engaged at the same time. Then you have a chance of actually selectively targeting AML. So the problem is how to do this, and there are several approaches that people are taking, but so far it's been a challenge. But that will ultimately improve the specificity of targeting and maybe the selectivity of targeting as well.